Model 23. X-linked inheritance and Y-linked inheritance pattern. The main objectives of this episode are to know about the sex chromosomes and the inheritance patterns, to explain about the X-linked dominant conditions, to understand the disease caused by the X-linked dominant genes, to know about the diseases caused by the X-linked recessive genes, to explain about the Y-linked inheritance pattern. Introduction. Chromosomes X and Y are the sex chromosome. Females consist of 2 X chromosome and male consists of 1 X chromosome, another Y chromosome. Since females have 2 copies of the X chromosome, and generally the gene product will be more compared to the males since they have only 1 copy of the X chromosome. To compensate the gene products with males, one of the X chromosome in females undergo inactivation is termed as the doses compensation. The disorders can be X-linked, where the mutations takes place in the genes of X chromosomes. Y chromosome is also the sex chromosome present in males along with 1 copy of the X chromosome. The many genetic researches involved in the identification of number of genes in each chromosome. It was found that the smallest Y chromosome consists of around 200 genes within the chromosome. Most of the genes are responsible for the production of proteins which show their active role in the male sex determinations and normal development. The genes residing on X chromosomes inherit differentially. Hence, in this episode, the following aspects are going to be emphasized. A. X-linked dominant inheritance. B. X-linked dominant disorders. C. X-linked recessive inheritance. D. X-linked recessive disorders. E. Y-linked inheritance. A. X-linked dominant inheritance. Dominant gene will mask the function of the recessive gene. In the case of X-linked dominant condition, the dominant gene will present in the X chromosome of the individual. In this case, both males and females are equally affected. Since it is a dominant condition, 1 copy of the gene is sufficient to show the symptoms of the disease. Males are in hemizygous condition, consist of 1 X and 1 Y chromosome, and the dominant trait of the gene will affect in case of males. Females comprise of 2 copies of the X chromosome, and in the dominant condition 1 copy of the gene expression is enough to get the symptoms of the disease condition. If an affected man marries an unaffected woman, then altered gene from the father will inherit to his daughters. The unaffected gene from the mother will inherit it to her sons. This shows that affected daughters will have the affected father. if a normal man marries an affected woman who is in heterozygous condition, then the possibilities of having affected son and daughter is 50% in the ratio 1:1. In heterozygous condition, one copy of the gene is mutant and the another copy will be normal. If the woman is homozygous, where both copies of the gene are affected by the affected gene, then all the children get affected, including son and daughter. Females are more affected than males in X-linked dominant condition. X-linked dominant disorder 1. Fragile X syndrome. This syndrome is mainly characterized by the problems in case of development, including intellectual disabilities. The affected child may suffer from the difficulties in speech and language development at the age of 2. This syndrome will affect the complete development of the child. More often, this condition is seen in males than females. A male child will suffer from severe symptoms, whereas the affected female child can suffer from intellectual disabilities. The gene gets affected in this condition is FMR1 and located in the chromosome XQ27.3 at the long arm of the X chromosome. This gene will encode for a protein called FMRP. The protein FMRP will play a vital role in the production of some other proteins and mainly helps in the formation of synapses. Synapse is the crucial connection between the nerve cells and they pass the impulses from one nerve cells to another cell. These impulses will reach the central nervous system in the brain and regulate every function of the human body. The mutations in the gene FMR1 will show the variation in the repeats of triplet nucleotide CGG. These repeats will lie within the FMR1 gene. In normal conditions, the number of repeats of CGG will be from 5 to about 30 times. The mutated gene will show the repeats of CGG about more than 200 times, and this will completely turn off the function of the gene. There will be no production of protein FMRP, and this will disturb the normal signaling of the nerve cells and affect the central nervous system. Some of the symptoms of this condition are intellectual abnormalities, anxiety and hyperactive nature, difficulties in managing the tasks, communication difficulties, a long narrow face with long ears, flat feet and flexible fingers, large testicles in case of males, and problems with hearing and vision. This condition is diagnosed during the pregnancy period for the presence of Fragile X. Some of the techniques like amniocentesis, where the amniotic fluid sample is taken for the test to check the changes in the gene responsible for the production of protein FMRP. Chorionic villus sampling test can also be done to check for the same gene by taking the samples of cells from the placenta. After the delivery of a baby, the blood test can be done to verify the presence of the gene FMR1. Treatment can be given in the form of some speech and language exercises and special education. There is no medications to cure this condition. In some cases, medications can be given to prevent some of the health issues. 2. Dermal hypoplasia. This genetic condition will mainly affect the skin along with bones, eyes, face development, etc. In the case of focal dermal hypoplasia, females are more affected than males. The affected males will show less severe symptoms. The affected gene in this case is PORCN. This gene located in XP11.23 at the short arm of the X chromosome. The gene is responsible for the production of a protein that modifies the other protein is called Wnt proteins. These proteins are the essential proteins which regulate the different proteins and help in the signaling pathway of the development of skin, bones, etc. The mutations in the PORCN gene will affect the production of PORCN protein. This will stop the release of Wnt proteins for the signaling pathway. This will mainly affect the normal development of the organ. The symptoms of these conditions are facial abnormalities like lip cliffs, small ears, abnormal teeth and pointed chin, etc. Eye abnormalities like a small size of the eyes, tear duct problems, abnormal development of retina can be occurred by these conditions. C. X-linked recessive inheritance. X-linked inheritance is the transfer of genes present in the X chromosome. In the case of X-linked recessive condition, recessive gene on the X chromosome will get inherited to the next generation. In this case, males are more affected. In recessive condition, the two copies of the gene should be affected to show the symptoms of the disease. Since female consists of two copies of the X chromosome and act as the carriers in heterozygous condition. Since males are hemizygous and get more affected than females. In case of recessive inheritance, they may show the skipping of generation in pedigree. If a normal man, that is XY, marries a carrier woman XX, then the chances of getting affected son and normal son is 50% in the ratio of 1:1. The chances of getting a carrier daughter and a normal daughter are 50%. Carrier daughter will not produce any symptoms. When an affected man marries an unaffected woman, then there will be no affected son produced. Best studied example for this condition is hemophilia. Carrier woman will transmit the trait for many generations. The affected male will never transmit the trait to his son. The affected father and carrier mother will produce the affected daughter. In case of the affected son, his parents are usually in affected conditions. Mother can be a natural carrier. X-linked recessive disorder 1. Duchenne muscular dystrophy, generally called as DMD. This is the group of disorder that mainly affect the skeletal muscles and degeneration of muscle cells in an organ like a heart. Skeletal muscles are essential for the movement. The gene affected in this disorder is termed as the DMD gene. This gene is located in XP21.2 at the short arm of the X chromosome. This gene encodes for the protein called dystrophin. Dystrophin is a large protein that connects the actin filament to the muscle cells and maintain the structural stability of the muscle fibers. This helps in the proper movement of the muscles. In the case of Becker muscular dystrophy, generally called as BMD, the same gene DMD is getting affected. The mutations occurred in DMD and BMD condition is different. Duchenne muscular dystrophy is the severe form and Becker muscular dystrophy is less critical. When the gene DMD gets mutated, it will result in the formation of abnormal or no protein production. Due to the lack of dystrophin protein function, muscles will not connect properly and become weak. In many cases, the muscle cells will start to degenerate and there will be the formation of fatty fibrous tissues. Some of the symptoms of this condition include unbalanced heartbeat, difficulties in breathing due to short breath, enlargement of the muscles of feet and legs, fatigues, tiredness, scoliosis condition where curved spines are seen, cramps and muscle pain and headaches. Diagnosis and treatment. Physical symptoms can be examined initially for the presence of this condition. Irregular structure of the skeletal system and other facial deformities will ensure the existence of this condition. Doctors also check the family history to ensure whether other family members developed the same disease. Muscle biopsy can be done by removing the small tissue and examining under the microscope to check the presence of protein dystrophin. Blood tests can be done to check the level of creatine kinase. The damaged muscles produce these enzymes. During the severe damage, there will be no increase in the level of creatine kinase enzyme. Genetic testings like checking the presence of the gene for dystrophin in an affected individual can be done. Along with that, females are tested for the existence of any mutated gene for this condition. This disease is under gene therapy. Hemophilia. This is a bleeding disease where the blood will not clot and continuous bleeding leads to severe issues. The normal condition shows the clotting of blood in place of any injury within 4 to 5 minutes. In the case of hemophilia, bleeding occurs continuously and the individuals mainly lack the clotting factors essential for the blood clotting. Depending on the clotting factor deficiency, there are two types of hemophilia. Deficiency in the clotting factor 8 results in the hemophilia A, and deficiency in the clotting factor 9 results in the hemophilia B. Both types will show similar symptoms and the mutation will occur in two different genes. Gene F8 located at XQ28 will code for the protein called coagulation factor 8. The gene F9 locate at XQ27.1 will encode for the coagulation factor 9. These two coagulation factors help in the clotting process during any injuries or wounds, and the blood clotting will not occur for more than an hour and results in continuous bleeding. In case of severe conditions of hemophilia, the complete function of these factors are lost. Red-green color blindness. This is a vision deficiency inherited in an X-linked recessive pattern. The affected people will face the problems in distinguishing between the shades of red, green and yellow colors. There are few genes get changed. They are OPN1LW located at XQ28, OPN1MW located at XQ28 and OPN1SW located at 7Q31.1 genes. These genes will code for the proteins, helps in the color vision. These two photoreceptor cells will transmit the signal of the visions to the brain. Rods will sense the low light and cones will sense the color as well as the bright light. These three genes will help in the formation of light sensing pigment in the cones. The defects in the genes will result in the abnormal condition of the vision. vision problems will occur along with some other symptoms like increased sensitivity of eyes to the light and back forth movement of eyeballs, etc. This can lead to the loss of vision. E. Y-linked inheritance. Y chromosomes are the sex chromosomes present in males. Y-linked inheritance is the inheritance of genes present on the Y chromosome to the next generation. Inheritance of trait in case of Y-linked inheritance will occur only in males. The affected male will transfer the trait to his son, and again sons will pass the trait to the next generation to their sons. Y-linked inheritance does not contain any dominant or recessive pattern. As mentioned, Y chromosome consists of few genes mainly responsible for the male sex determination. SRY is one such important gene, acts its role in the development of a fetus into a male. Few other genes are responsible for the different traits and male fertility. There is also the presence of the pseudo-autosomal regions, and genes present in those regions are responsible for the normal development. The hypertrichosis of the ear is the trait of having hair on the ear pinna, and the webbed toes are some other characters of Y-linked inheritance in humans. Holondric genes are the Y-linked genes responsible for the transmission of trait in males. Holondric gene consists of one copy of the allele. There are many genes responsible for the sex determination functions like sperm development, hormonal control in males, and development of secondary sexual characters. To conclude, depending on each gene mutation on X chromosome, the different disorders are identified. Due to the practice of consanguineous marriages and inbreeding within the community, the recessive genetic diseases are seen more in South India. Thank you.